Good day from Chem Help ASAP. This video describes the recently approved drug RITLECITINIB sold under the brand name of Litfulo. This video emphasizes the medchem properties of the drug rather than the clinical pharmacology. If you happen to be a scientist who worked on this project and are at liberty to speak about this research or your general work at Pfizer, I will be very interested in interviewing you for a separate video. The channel email address is in the video description. The approval of RITLECITINIB's NDA was granted on the 23rd of June in 2023. The active ingredient is RITLECITINIB, which is the U.S. adopted name. The branded name of this drug is Litfulo. RITLECITINIB is an oral drug and it is administered orally as a 50 milligram daily dose. RITLECITINIB's indication is for the treatment of severe alopecia areata in adults and adolescents 12 years of age or older. Alopecia areata is an autoimmune disorder that results in a patchy form of hair loss that is distinct from conditions like male pattern baldness. I believe it is considered to be a rare disease. Pfizer sponsored the NDA. All this information can be found through the prescribing information of RITLECITINIB through the link at the bottom of the screen. RITLECITINIB, which has a research designation of PF 066-51600, has been studied in a large number of clinical trials. The indications for those trials range from alopecia areata to Crohn's disease, rheumatoid arthritis, and ulcerative colitis, as well as a number of PK studies, many of which monitored potential drug-drug interactions. RITLECITINIB's two listed targets are JAK3, Janus kinase 3, and TEC, tyrosine protein kinase. JAK3 is expressed primarily in immune cells and within the intestinal epithelium. TEC is widely expressed, including in B and T cells. Inhibition of both these targets is connected with immune disorders, and all the indications listed for the clinical trials are indeed immune disorders: rheumatoid arthritis, ulcerative colitis, Crohn's, and of course, alopecia areata. There are existing inhibitors for both JAK3 and TEC. On the left is tofacitinib, which is approved for both rheumatoid arthritis and colitis. You can look up the IC50 of tofacitinib for JAK3 in IUPhar's Guide to Pharmacology. See the link below. And the reported IC50 values range from 1 to 200 nanomolar. That's quite a range, but IC50 values do vary depending on how the assay is performed. Ibrutinib is a TEC inhibitor with an IC50 of 8 nanomolar. TEC is not the main target of ibrutinib, but ibrutinib is an established TEC inhibitor. Notably, ibrutinib does also have reasonable activity on JAK3 as well. Where does RITLECITINIB fit within these two compounds? RITLECITINIB has an IC50 on JAK3 of 90 nanomolar and TEC of 532 nanomolar. These data come from a JMedChem article. The link is below. Specifically, the data come from Table 6 of the article. I have also listed the Ki values for RITLECITINIB for these two targets. For reversible competitive inhibitors, Ki is normally smaller than IC50. That's not the case with RITLECITINIB because RITLECITINIB is not a reversible competitive inhibitor. This compound is an irreversible inhibitor. It forms a covalent bond to both targets, JAK3 and TEC, and we'll discuss the mechanism of inhibition in a couple slides. Regardless, the point of this slide is that RITLECITINIB binds to both JAK3 and TEC, which are targets associated with immune suppression. Furthermore, RITLECITINIB has structural similarities to other known JAK3 and TEC inhibitors. Here are some physicochemical parameters for RITLECITINIB. The compound is a small compound with a molecular weight of just 285. The log P, hydrogen bond acceptors, and hydrogen bond donors are all quite low for a drug-like compound. In fact, these properties are more lead-like than drug-like. Of course, the binding of RITLECITINIB for its targets based on Ki in the micromolar or barely submicromolar range is also rather lead-like, not drug-like. How is RITLECITINIB a drug? Well, drugs are not approved based on their binding. Drugs are approved based on being safe and effective. Despite RITLECITINIB's somewhat modest binding, the fact that it covalently binds its targets helps boost its efficacy beyond what you'd expect based on its binding affinity alone. Let's visualize this binding and reaction. Here is a screenshot from an entry 5-TTU from the protein data bank. This structure was deposited by scientists at Pfizer and is included in the previously mentioned JMedChem article. This is not RITLECITINIB, but an analog, compound 7, in the paper. The two structures are very similar. In the structure, the aromatic heterocycle is buried in the binding pocket in the lower left. That places this acrylamide feature adjacent to a cysteine residue. So on structure 7, here is the alpha carbon and here is the electrophilic beta carbon. Let's label these same atoms in the X-ray structure. The nucleophilic sulfur is right near the beta carbon. In a Michael addition, a covalent CS bond forms. The inhibitor is now covalently bound in the active site, and this kinase is rendered inactive, no longer able to bind its normal substrate, ATP, for the phosphorylation of proteins. Because covalent binding is a great way to permanently render a target protein inactive, irreversibly binding drugs are very attractive. A major challenge, however, is making sure that the drug only reacts with the intended target, or targets in this case. The drug should not react with other targets. The drug must also be sufficiently inert that it circulates for some period of time to achieve efficacious exposure in the patient. This is a hard balancing act. Michael acceptors, like the one in this structure, have emerged over the past 15 years or so as a viable way to design covalent inhibitors for various kinases. On to PK, here are the key pharmacokinetic parameters for RITLECITINIB. Although dosed just once a day, the drug has a fairly short half-life. A short half-life is often not ideal for once-daily dosing, but the irreversible binding of RITLECITINIB allows the drug to achieve its task. The patient takes a dose of the drug. The drug circulates long enough to bind and disable some fraction of the target protein in the patient. The remaining drug is quickly eliminated. Despite not having a lot of drug present, the patient enjoys the benefit of the drug until his or her body makes more target protein. Making new proteins takes many hours, so the patient gets the benefit of the drug without having prolonged or extended drug exposure, which can pose safety risks. So even though the PK of this drug is short, based on the half-life, the PD, the effect is much longer and amenable to once-per-day dosing. The other PK parameters are unremarkable: moderate clearance and distribution, good bioavailability, low plasma protein binding, and very little of the parent drug is excreted unchanged by the kidneys. Let's discuss RITLECITINIB's metabolism just a bit. There are multiple routes of metabolism for RITLECITINIB, both phase 1 and phase 2 reactions. According to the prescribing information, no single pathway accounts for over 25% of the overall metabolism. With this Michael acceptor handle on the left of the structure, RITLECITINIB is somewhat prone to undergo glutathionylation. So, just as the parent drug can react with the thiol in the target protein, the parent drug can react with the thiol group of glutathione, GSH. Once conjugated with glutathione, the metabolite will be quickly eliminated. Let's close with safety risks. The two items on the screen are in a black box because RITLECITINIB does carry a boxed warning from the FDA. As RITLECITINIB is an immunosuppressant, patients taking the drug are at a greater risk for viral, bacterial, and fungal infections. Patients must also avoid using RITLECITINIB with other JAK inhibitors and TNF, tumor necrosis factor blockers. Taking these compounds together puts the patient at risk for all sorts of issues, including cancer, all-cause mortality, and MACE, major adverse cardiovascular events. Those are some highlights for RITLECITINIB, marketed as Litfulo. While RITLECITINIB is currently approved for severe alopecia areata, based on its many uses in clinical trials, I would expect that Pfizer might file additional NDAs for other indications in the future. Thank you for watching to the end of this drug approval summary for RITLECITINIB. Please consider subscribing to the channel, leaving a like, or making a comment. Take care.